Okay, for part 2 of chapter 15 on translation, we're going to look closely at how the genetic code is read. Here's a strand of DNA at the top here in purple. We have 2 strands of DNA. DNA is double-stranded. It has a coding strand and a template strand. Remember that the coding strand has the same nucleotide sequence as the messenger RNA, while the template strand is complementary to the messenger RNA that's produced. Here's the sequence of the messenger RNA. Note that its nucleotides are A, C, and U substituting for T and G. When you're reading a code of messenger RNA, the first thing you need to look for is the start codon. All protein synthesis starts with an AUG at the 5' end. Okay? The start codon is complementary to the anti-codon of the tRNA. The tRNA brings in an amino acid, one amino acid for each codon, and the anti-codon portion of the tRNA is complementary to the codon. So an A will base pair with a U will base pair with an A, and a G will base pair with a C. The start codon sets the reading frame for the rest of the sequence of the messenger RNA, of the protein. Each triplet codon that follows encodes one amino acid until you reach the stop codon, which doesn't encode any amino acids. Flanking the coding region of the messenger RNA is the 5' untranslated region and a 3' untranslated region. Note that the start codon sets the reading frame for all the remaining codons. There's the start codon right there. So here is an amino acid chain that has been synthesized based on this sequence of messenger RNA. Notice that there are gaps in here in between the triplet code. That does not exist in a messenger RNA. All of these nucleotides are right up against each other. They did this just so that you can understand that 3 nucleotides equals 1 amino acid. So this slide illustrates the fact that the polypeptide has an amino terminal end and a carboxy terminal end, and that the amino terminal end always corresponds to the 5' end of the messenger RNA, and the carboxy terminal end always corresponds to the 3' end of the messenger RNA. Here's the amino terminal end and the 5' end, and the carboxy end and the 3' end. So when you have finished synthesizing a polypeptide, adding one amino acid after another, you end up with a long chain of amino acids that is the protein, with the amino terminus corresponding to the 5' end of the messenger RNA and the carboxy end corresponding to the 3' end. This amino acid chain is 120 amino acids long. This polypeptide is 120 amino acid long. How many nucleotides does that represent? You should definitely be able to figure that out. Very simple math. We're not going to talk in great detail about this, but file this information away for later on when we do start to talk about point mutations and how they affect protein synthesis and whether or not those mutations end up being dominant or recessive. There are different levels of protein structure, are primary, secondary, tertiary, and quaternary. Primary structure just refers to the sequence of amino acids. The next secondary structure is how those amino acids fold up initially, either into an alpha helix, a very common structure, or a beta sheet, also a very common structure. And when you take biochemistry, you'll learn a whole lot more about that. The tertiary structure of a protein is putting the alpha helices and beta sheets and all the bends together into a globular type protein. And quaternary structure refers to the fact that many proteins are made up of subunits that come together. Okay, so here's a molecule that you should be familiar with. This is a tRNA molecule. The tRNA is the adapter molecule. In the 1950s, Francis Crick, after they had figured out the structure of DNA, after he and Watson had figured out the structure of DNA, and Matty Hoagland proposed the adapter hypothesis. They knew that genes encoded proteins, so there had to be some kind of translator, something that would turn the information stored in the genetic code into amino acids. And that molecule turned out to be tRNA. This is the three-dimensional structure of a tRNA, and this is the flattened out two-dimensional version. tRNAs recognize the 3 base pair codon in the messenger RNA, and they carry an amino acid that's specific for that codon. tRNAs are named according to the amino acid they bear. So if there are several different codons for phenylalanine, there will be several different tRNA phenylalanines in the cell. Here's an example of what a tRNA needs to do. tRNA needs to bring the right amino acid to the messenger RNA. And the way it does it is by hydrogen bonding between the anticodon of the tRNA and the codon of the messenger RNA. Note that there's this artificial space introduced in here as well. Proline, here's another tRNA for proline. It has a different anticodon to hybridize to the different codon. Okay, notice that these molecules are nucleic acids. They are both RNAs and they have directionality. And just like DNA, the 2 strands of DNA, when they hybridize to each other, when they hydrogen bond to each other, they are anti-parallel. The 5' end of one strand is opposite the 3' end of the other strand. The same is true for tRNAs and messenger RNAs. Here's the 3' end of the tRNA, of the anticodon. That's opposite the 5' end of the codon. Okay? And the 5' end is on the other end. tRNAs have a universal structure. They all have to be able to fit into the same slots in the ribosome. So they all have a very similar universal structure with some slight variations. All tRNAs at their 3' end have the sequence ACC. That is called the acceptor stem, and amino acids are covalently attached to the adenine at the very tip of the 3' end of the tRNA. And there are special enzymes in the cell that accomplish this. You'll notice that in the sequence of tRNAs here, there are some modified bases. This occurs after the tRNAs are synthesized. Inosine, methylinosine, etc., etc. There are special modifications. They help to stabilize the tRNA and protect it from degradation in the cell. Okay, what about these enzymes that attach the amino acid to the acceptor stem of the tRNA? The enzymes that attach amino acids to tRNAs are known as amino-acyl tRNA synthetases. These are by far my favorite enzymes in the whole cell. There are 20 types because there are 20 different amino acids. Synthetases catalyze a 2-step reaction involving 3 different molecules: the amino acid that they charge, tRNAs, and an ATP. So here's our synthetase here. Notice they have a little slot here for ATP and a slot here for an opening for the specific amino acid. Say this is an alanine tRNA. Okay, so alanine will come in here, and then a spot here for the tRNA. The first thing that happens is that the amino acid and ATP bind to the enzyme. AMP is covalently bound to the enzyme and pyrophosphatase is released. So this is a covalent attachment of AMP to the amino acid, okay? And pyrophosphate is released. Many times when ATP is used in a reaction, it is not covalently attached to one of the substrates, but in this case it is. Okay, the next thing that happens is that the correct tRNA binds to the enzyme. The amino acid becomes covalently attached to the tRNA. Okay. And the AMP is released. And finally, you release the charged tRNA. One of the reasons that this is one of my favorite enzymes is because not indicated in this diagram, there's a lot of proofreading that goes on during this process to make sure that this tRNA is the correct tRNA for this amino acid. One of the other reasons that I really like this enzyme is because this same enzyme for alanine will accommodate several different, well, 3 or 4 different alanine tRNAs in this slot. Only alanine here, but different tRNAs for the alanine here. And they'll all get charged correctly with the correct amino acid. Okay, so now we refer to this tRNA that has an amino acid covalently attached at the 3' end as a charged tRNA. This is an idiosyncrasy of the codon, the genetic code, that Francis Crick recognized after the genetic code was published in 1966. In the codon anticodon recognition process, the first 2 positions pair strictly according to the AUGC rule. In other words, A always pairs with U and G always pairs with C. That's for the first 2 positions. But the 3rd position can actually wobble or move a bit, and it tolerates certain types of mismatches. But it turns out that those mismatches in the 3rd position don't really matter too much. Let's look at proline. Okay, these are the 4 different codons for proline. Notice that the first 2 are always CC. CCU, CCC, CCA, CCG. So if by chance the wrong tRNA gets in, binds correctly to the first 2 nucleotides, but the 3rd one is a little off, it doesn't matter because you're always going to have a proline inserted into that slot. And if you look through the codon and follow the rules of the Wobble base pair hypothesis, you'll find that very, very seldom would a wobble in the 3rd position actually introduce an incorrect nucleotide. Okay, let's look at some more of the genetic code while we have it up here. Notice that there is one start codon, and that start codon always encodes the amino acid methionine. And I'm going to tell you an exception to that rule when we talk about our phage genome annotation project. But for our purposes right now, AUG is the start codon and it always encodes methionine. And then there are 3 stop codons, UAA, UAG, and UGA. And none of these encode any amino acids. All right. So what about this wobble position? Here is our friend phenylalanine tRNA. In the first position of the codon, we have a U. In the second position, we have a U. In the 3rd position, we have a U as well. This is depicted as having the correct base pair for the first position and the second position, AU, AU, but the 3rd base pair is not the right normal conventional base pairing. Okay? What the Wobble hypothesis says is that this nucleotide of the anti-codon, the Wobble base of the anti-codon, can actually shift somewhat, a little bit, so that it can hydrogen bond with a couple of different nucleotides, a U instead of its normal G. And that's what we see depicted here. If this C is in the Wobble position here in the anti-codon of the tRNA, it only ever always pairs with a G. If, however, this nucleotide is an A, it can base pair with everything, even sometimes with itself. And if it's a U, it can also pair with everything. And if it's an inosine, which is a modified base, it can pair with these. Okay, that's it for part 2, and we'll see you in part 3.